Is it showing up or no? Are you able to see my screen?
Yes, we are.
Yes.
Okay. Okay, good. Because I don't know if you're seeing my screen or not. Okay. Okay, can you tell me what organ this is?
The thymus?
Thymus, very good. Okay, because we have lobes. Okay, it's a lobulated organ, and it has this outside region called the outer dark region called the
Cortex?
cortex, and the inner paler region called the
Medulla?
medulla. Okay, so is this like a primary lymphoid organ, a secondary lymphoid organ? What do you think?
Primary.
Primary. Okay, and what kind of cells mature here?
T cells?
T cells. What name do we give them? What do we call those maturing T cells?
Curing T cells?
Yes, the T cells that are in the thymus. What do you call them?
Naive T cells?
They have a specific name. Naive T cells is still general, but the T cells in the thymus,
T lymphocytes?
they are all lymphocytes.
Lymphocytes.
The ones in the thymus are called thymocytes.
Thymus. Okay.
Okay. So the thymocytes come here, T cells come here from the bone marrow and they reside in the thymus. And there we call them thymocytes. Okay. And they undergo maturation. And then they get back into the circulation. Okay. So when they are sitting here in the thymus, okay, all these are lymphocytes or thymocytes. Okay. You see lots of them. But in between, let's go 40x. Let me go to a good region. Look at that guy there, okay, with a large pale nucleus. Okay, let's go. Okay, like right there. Why can't I zoom in beyond this? I don't know why I can't, but let's go keep looking because there are lots of them. These thymocytes are there only on a temporary basis. Okay, look at this guy here. Okay, euchromatic nucleus and lots of pale cytoplasm around it. Okay, so that forms the parenchyma or the stroma of the thymus.
The stroma?
The stroma, okay? Because this is like the scaffold. See, look at that one there, big, pale nucleus. So that's an example of a stromal cell. So what are these stromal cells called in the thymus?
Epithelial reticular cells?
Awesome, epithelial reticular cells. What do they produce? What kind of fibers do they produce, first of all?
Reticular fibers?
Reticular fibers, okay? Can you see them with H&E stain?
No.
No, you cannot. So you have to have special stains to see those fibers. Usually silver stain works the best, okay? So epithelial reticular cells, they produce very little fibers actually. There should be other reticulocytes that are producing the fibers in the thymus, okay? But they produce a hormone. What is that hormone called? Thymopoietin is the hormone, okay, that the epithelial reticular cells produce. Okay. Okay, so let's zoom out a little bit. So these epithelial reticular cells, where do they originate from? Embryologically, where do they come from?
The ectoderm?
From the ectoderm. So it comes, so because of that, they go back, they try to kind of revert back to their embryological origins from time to time, you know. So they become, look at that, that's why they called epithelial reticular cells, okay? And they try to become stratified squamous, produce keratin and all of that. So that's why you see these worlds, okay? In the medulla, you can see them especially in older animals. Okay, so what are these structures called?
The thymic corpus.
Thymic corpuscles or Hazell's corpuscles. Okay, so that is what we call them. So here is a good example. There is another example. Here's another example of one that is in process. Okay, so you have to know all the different types of lymphocytes like that we talk about the lecture notes that Dr. K talked about. So you have to know the different kinds of lymphocytes that are present and where they come from, you know, and what happens to them. So they come here to the thymus, they mature. So who are all these like cells who are sitting in these spots that look like a starry night, right? Stars in the sky, all these spots. What do they contain?
Macrophages.
Macrophages, very good. So those are the different cell types that are present in the thymus. Okay. So how do these thymocytes come in? They come from the bone marrow, but how do they get into the thymus?
The
Through
lymph
the blood
system. The
circulation.
lymphatic system.
No, not through the lymphatic system. They come in through the blood circulation because lymph does not percolate through the thymus. It percolates only through the lymph nodes. Okay, so this, so it comes in through the blood vessels. So you have all these blood vessels, right? There are capillaries. Okay, how do we know that these are capillaries? Because there is only a single endothelial cell lining this. Okay, so these are all capillaries, and there are like 3 different types of capillaries, right? Do you remember?
Fenestrated, continuous, and discontinuous.
Discontinuous, awesome. So the continuous capillaries are going to be present in the cortex, okay, but if you come into the medullary region, you'll see a lot of fenestrated capillaries. So these have holes in them, so that is how these lymphocytes escape, okay, and they populate the thymus. So that is the thymus for you. So let's go to another lymphoid organ. Immune and we have to go to the immune system first. Okay, now what is this an example of? What organ is this?
So the spleen.
This is, okay, spleen is this guy.
Okay.
So this is the spleen, okay? There is another section of the spleen right here. See, it's a solid organ, okay? It has a capsule. It has these nodules, okay, that are kind of distributed all over. And you can see the trabeculae coming in, but it's mainly divided into red pulp and
White pulp.
white pulp, okay? So that is typical of the spleen. So if you look at this structure or this structure, you can see the red pulp, right? That is red, and all this blue is actually white cells. So we call it the white pulp. So if you cut like a spleen in gross anatomy, you will be able to see specks of white. Okay, so that is the white pulp. The rest of the area will be red, so we call it the red pulp, literally. Okay, so if you go to this structure right here, this is a lymph node. Okay, so that is one lymph node. Okay, This is another lymph node. This is another lymph node. Okay. So how do we call this the lymph node? Because it has this outer region called the
Medulla.
outer region is always the
Cortex.
cortex, and the inner region is the medulla. This is a weird cut. Okay, so it looks like the medulla is on the outside, but the cortex is on the outside, medulla is on the inside. Okay, but the cortex has these round structures called the
Nodules.
nodules. Okay, so these are lymphatic nodules. And lymphatic tissue, like wherever you see lymphatic tissue, you will see these nodules except for the thymus. Okay, all other lymphatic tissue will have these nodules. So a typical nodule has parts, right? So let's go closer to look at this a little bit. Let's go to the cortical region. Okay, let's say that this is a nodule. So here is a nodule, there's a nodule, there's a nodule, there's a nodule, right? So the nodule has an outer zone and an inner zone. Okay, so the outer part is called the,
Mantle.
yes, the mantle zone, and the inner part is called the,
Germinal center.
yes, so that's the germinal center. Okay, so the germinal center is always going to be pale in color. Okay, you have more mature cells in the mantle zone, you have more immature cells, dividing cells in the germinal center. Okay, and this is rich, this lymphatic nodule is rich in what kind of lymphocyte?
B cell.
B cells. Okay, and the paracortical zone, which is the space in the cortex that is surrounding these nodules, see all the space in between the nodules, we call that the paracortical zone. Okay, that is going to be rich in
T cells.
T cells. Okay, so you have B cells in the nodules and T cells in the paracortical zone. Okay, so let's look at, so this structure here is the
Capsule.
capsule. Okay, so the capsule will send like projections down into the substance of the lymph node, and these are called
Trabeculae.
trabeculae. Very good. So the trabeculae extend from the capsule down into the substance of the lymph node. Okay, so there is a sinus under the capsule. Okay, into which blood vessels that drain into these lymph nodes enter. Okay, so what are the lymphatic structures that enter the lymph node called? They come from the surrounding tissue. Okay, so the lymph node acts like a local police station. So all the roads from the surrounding lymphatic surrounding tissue
The
get
hilus.
access. So, yes.
Is it the hilus?
No, hilus is the place where the lymph vessels take stuff out of the lymph node. Okay, so this is input. These are vessels that bring in lymph from the local region to this lymph node. So what are they called?
Afferent levels.
Afferent.
Afferent lymph cells. A, okay, because they come to the lymph node. And the ones that leave the lymph node are called
Efferent.
efferents. So for example, see all these are efferent lymph cells. Okay, but you will have afferent lymph cells coming in here. We don't have like too many good examples, but maybe in another section we can spot some. Okay, so the afferent lymph cells bring in the lymph and it filters through the subcapsular sinus. Okay, like right there, and then it could pass along the trabeculae through the trabecular sinus, okay? And then it filters through the cortex and comes down to the medullary region, okay? So the medulla of the lymph node has two distinct regions, okay? These darker regions are called
Cords.
medullary cords, very good. And what are these paler regions called?
Sinuses.
Medullary sinuses, okay? So you have to go back to the capillaries again. So we had continuous capillaries, right? We had fenestrated capillaries, and then we had discontinuous or sinusoidal capillaries, right? So these sinuses are actually those kind of blood vessels, sinusoidal blood vessels. So they're going to be lined by endothelium, okay? So these are all sinuses, okay? Relative to the cardiovascular system, the lymphatic system, they're all going to be lined by endothelial cells. So these guys, the sinuses, contain what? Let's go take a look at it. You can see lots of cells, right? So there are lymphocytes again, but you can also see some of the stromal cells, you know, like the ones here. Look at that. So it has like projections, like it's dancing. So this is forming the stromal network for the lymph node, okay? Again, the fibers here are going to be
Reticular fibers.
reticular fibers, okay? You see lots of reticular fibers, and all these are reticular cells, okay, that are producing these fibers and forming the stromal network. You can also see some cells that have a marginated chromatin, okay? These are big cells that have a nucleus that has a cartwheel kind of appearance. See, like these big purple cells, you know, those guys are, they're not like round, tight looking lymphocytes. See, like this guy here, purple looking with like eyeliner kind of a nucleus. So all those guys are called like that one there. Those are all plasma cells, okay? So plasma cells produce what?
Antibodies.
Yes, they produce antibodies, okay? So they reside here on the medullary cords and they keep producing antibodies, which can also be sent out into the general circulation or through the efferent lymphocytes. What are these cells that contain these brown cytoplasmic inclusions? Who are they? These guys.
Macrophages.
Macrophages. Okay, so these are all macrophages that are also here in the lymph node. Okay, so if we zoom out a little bit, let's zoom out. There are two type of lymphatic nodules. Okay, so there are, what are they?
Primary and secondary.
Yes. So looking at these, would you call these as primary or secondary?
Primary.
So primary will be generally dark, okay? The secondary are the ones that will have this germinal center and the mantle zone. These belong to activated lymphatic nodules, okay? Because they are actively generating new cells that are ready to fight against whatever invasion that the lymph node is taking on. So these are all secondary nodules. So that would indicate that this lymph node is being activated. Okay, if it was a primary lymphatic nodule, it would be quiet and dark, like maybe this one. Maybe we are cutting the edge of the lymphatic nodule here, but if it was not activated, this is how it would look. Okay. So that is it for this lymph node. So
Dr. Eva, I actually had a question for the lymph node. Can you
okay.
see the antigen presenting cells?
You have to have stain them with specialized stains, with antibodies to actually differentiate the antigen presenting cells. You won't be able to see them in an H&E stain distinctly.
Got it. Thank you.
We can say, okay, there are antigen presenting cells here, but we won't exactly know who it is unless we use specific antibodies. So let's look at this lymph node. So here, I think this one is from a pig. Okay, the medulla looks like it's on the outside, and the cortex looks like it's on the inside. That's because that's how pigs are. Okay, their arrangement is kind of inversed or reversed. So let's look at this one. This is another trichrome of another lymph node. Here you can see the trabeculae coming in. You can see the subcapsular sinus. Okay, let's go closer. See, that's the subcapsular sinus. There's the trabecular sinus, okay? And all these medullary cords and sinuses in the medulla, efferent lymphatics coming out, okay? All those are efferent lymphatics coming out. So same structures, but this is a much quieter lymphatic nodule. You don't see too many activated nodules here. Okay, so this is a quieter lymph node compared to this one here, which is more reactive. Okay, so that's the lymph node. So now let's go to the spleen. Okay, so the spleen has two zones. What are they?
Stroma and parenchyma.
Stroma and parenchyma make up the spleen, but it's not like zones, right? It's like all over. So what is the difference between stroma and parenchyma?
Stroma is the active part of the organ.
I can barely hear you, sorry.
I'm sorry. Is it something about the stroma is more of the active part of the organ?
Okay, so think of it this way. Okay, stroma is the structural scaffolding of the organ. Okay, so stroma structure, think of it that way. Okay, both of them start with an STR. The parenchyma is actually the functional component of the organ. Okay, so all the cells that produce something or participate in the function of the organ form the parenchyma. So here the stromal cells would be the reticular cells, okay, that provide the structural framework for these lymphocytes, which form part of the parenchyma, okay, lymphocytes in the macrophages. So they form the parenchyma here. So if you look at the spleen closely, we have two distinct zones, right?
Mm-hmm.
What are they? Two distinct areas.
The
The red pulp and the white pulp.
Exactly. So the red pulp is here, okay, and the white pulp is all this blue stuff, which in real life will look white, okay. So the blood, when it comes in through the splenic artery, see, like this, okay, when it comes in, and these are all probably branches of the splenic artery. So when it comes in, and some of these branches are coming at you, okay. So these branches are going to be covered by lymphocytes, okay, like a lymphocyte covering right here, lymphocyte covering around this vessel, around this vessel, around this vessel. See, around this big vessel here. So what do we call this lymphocyte covering this vessel?
ellipsoids.
Mm-hmm. It is called, okay, I'll tell you, there is the name of there is an artery in the name. Okay, here is a good one. Here is a blood vessel coming in and it's covered by this.
It's the artery of the white pulp.
Artery of the white pulp is this, but what is this covering of lymphocytes called?
The periarterial lymphatic sheath.
Yes, that's the periarteriolar lymphatic sheath, or the pals. Okay, is what we call them. So they cover these blood vessels. Okay, they sheath these blood vessels. Okay, so that's why we call them periarteriolar lymphatic sheaths. Okay, so it could just be like a covering of white blood cells, but it can also have a nodule associated with it. You see? So here is a small blood vessel in cross section. You have the pals right here. Okay, but there is a nodule right here, which has the mantle zone in the germinal center. You see, just like in the lymph node. So here is another blood vessel. The pals is right here, but the nodule is right here. Can you see it?
Mhm.
So you have the pals and the nodules, and both of them contribute to the white pulp. Okay, so the white pulp is made up of nodules and the pals. Okay, so what is this artery called in the middle, or arteriole?
Central artery.
Central artery, central arteriole. Okay, and sometimes the central arteriole will branch off, right? So it will branch off, it will give rise to two arterioles, and finally it will become
Capillaries.
capillaries ultimately. Okay, but before they become capillaries, there are like a few of them. Okay, like right there, these are called peninsular arterioles. Okay, so from there what happens, so the blood that comes through these arteries, it flows through the arterioles, through the peninsular arterioles, and then it kind of goes through capillaries that diffuse here and into the red pulp.
Red pulp.
Okay, so this zone that surrounds the white pulp is called the, like on the very edge,
Splenic follicle.
it's called the marginal zone. Okay, so it's called the marginal zone on the outside. So the blood goes through the marginal zone and then it goes into the red pulp. Okay, so there are capillaries all the way around. Okay, and from these capillaries it gets into the sinuses, which are also capillaries, right? Sinusoidal capillaries of the red pulp. Okay, so from there, this is the capillary bed in the red pulp. Okay, so from the arterial end, the blood will flow into the venous end, and then it will flow into venules. Okay, and then it will flow into larger veins, and ultimately it will exit through the splenic vein. So that is how blood flows through the spleen. Okay, so when you study the lymph node, know and understand how lymph flows through the lymph node. And when you're studying the spleen, know and understand how blood flows through the spleen. Okay, because it's very important. And the way the blood filters through the red pulp, it kind of slows down when it goes through the red pulp, and it provides an opportunity for the macrophages that are in there. So let's go look at some macrophages. See, all this is sinuses, okay? And all these dark brown staining cells that you see there, who are they?
Splenic macrophages?
Yes, macrophages. So they are sitting there in the splenic cords, like the medullary cords and sinuses that you have in the lymph node. you have splenic cords and sinuses. Okay, so they are sitting in the splenic cords and they are capturing all the dying red blood cells that are flowing, that are filtering through the space. They are capturing them and they are digesting them and they are retaining just the iron part. Okay, iron is a very precious commodity in the body. You can't like always get it from the environment. So the body kind of tries to recycle as much iron as it possibly can. Okay, so that is why these macrophages are saving all this iron, and that's why their cytoplasm is brown. So there is a specific name for this iron pigment. Okay, you will read about it in pathology also. It's called hemosiderin. Okay, so that is the name of the iron pigment that is present in these macrophages. So let's go. Same thing, lymphatic nodules here, pals here, central arteriole right here. Okay, so that is it about the spleen. So this structure here is the bovine spleen. Okay, here again you can see a big thicker capsule. Okay, look at the capsule. See how thick it is. All this is smooth muscle, by the way. Let's go take a closer look at this. See, all these are smooth muscle fibers. Can you see them? And then there is the capsule with collagenous fibers on the outside. See, there is collagen, and then all this is smooth muscle. Okay, and this is from a bovine spleen. So here is a trabecula extending with lots of smooth muscle in it. Okay, and it's kind of difficult to see the red and white pulp. Okay, all this is white pulp. You can see it, right? All this is white pulp and all this is red pulp. Okay, but here the distinguishing feature is the presence of these capillaries in the marginal zone or even in the sinusoid. Look at this, that are encapsulated. See, like here, these are capillaries, okay, but they are covered by macrophages that are sitting on top of them, waiting for these dying red blood cells to come out. So that's an ellipsoid right there. There is an ellipsoid. Okay, look at that. There's an ellipsoid. So you just have to go around hunting for them. Okay, and once you become good at identifying them, those are the sites where macrophages are sitting, ready to engulf the dying red blood cells. Okay, so ellipsoids you'll be able to see only in the spleen, bovine spleen. You won't be able to see it in the other spleen very nicely. Okay, so that is it. Do you have any questions about the spleen and the lymph node? Okay, one more thing. Okay, so there are here again, when you look at the blood vessels, okay, there are blood vessels that come into the lymph node too. It's just not the lymphatic vessels that bring in lymph. There is actually blood circulation through these lymph nodes also. See, like right there, that's a blood vessel. You can see the blood cells in there. See, blood vessel, right? So, but if you look at the endothelium, it's not, the nuclei are not flat like you would see in a normal endothelium. See, the nuclei of these endothelial cells are more prominent. And these endothelial cells are not looking like squamous cells. They look almost cuboidal. Okay, so we call them high endothelial vessels. Okay, so the lymphocytes tend to escape out from the blood circulation through these high endothelial blood vessels. So that's why you should know that these lymphatic organs have these specialized vessels with the specialized epithelium. High endothelium is what we call them. See, the endothelial cells are kind of more prominent. They don't have like a flat nucleus squamous cells, but these squamous cells are a little taller. Okay, so high endothelium is what we call them. Okay, so let's go back to some more immune tissue. Okay, so where do we find the rest of the immune tissue? So all these, the spleen, the thymus is like an isolated immune organ, primary lymphoid organ. Spleen is a lymphoid organ. Then you have lymph nodes that are many, right, distributed all over the body, different regions. And then you have localized immune tissue. Okay, so for example, hey, what is this? Okay, localized immune tissue, right? So in specific regions, like these, for example, this is an example of a
Liver? No, lymph node.
no, it's not a lymph node. It looks like a lymph node, doesn't it? But it's like a regional lymphoid tissue.
The tonsil?
Tonsil.
Tonsil. How do we call this a tonsil? You have to look at the epithelium associated with it. Okay, so if you see this, oops, sorry. This epithelium on one surface, let it kind of there. What epithelium is this?
Stratified squamous?
Yes. So this is stratified squamous, and this is probably present in the oral cavity. Okay. So this could be a palatine tonsil or a lingual tonsil or a pharyngeal tonsil, something like that. Okay. So all these areas are lined by stratified squamous epithelium. So you have stratified squamous epithelium associated with this lymphatic tissue. Okay, so you can see nodules, okay, that are characteristic of lymphoid tissue, lots of nodules, and then you have this epithelium. So once you have this, then you can safely call this a tonsil. Okay, the tonsils sometimes have crypts, okay, that are just invaginations of the surface epithelium to increase the surface area. Okay, so we call them these spaces crypts. Okay, so that is one tonsil. Here is another tonsil. See, you can see a nice crypt going in here. Okay, again you can see lots of nodules. This is probably like a activated tonsil because you're seeing secondary lymphoid follicles. Okay, and if you go closer to the surface, you can even see this epithelium, which is
Stratified squamous.
yes, stratified squamous. Is it keratinized or non-keratinized?
It is not keratin.
Non-keratinized.
Not keratinized. Very nice. Okay, so there was another. So here you can see lymphoid follicles again. Okay, and if you see these white spots, what does it mean?
Are they macrophages?
Yes, those are all macrophages that are sitting there. Okay, so this is a nice looking tonsil. And here there was another interesting thing that Dr. J pointed out. So this is like a granuloma right here. You'll learn about that in pathology, okay? But let's not worry about it right now. So all these are lymphatic nodules associated with this tonsil, okay? This structure here, what is this?
Glands?
Yes, this is a gland. What is it secreting?
Mucosal glands? So
Yes. So see, what is it producing? Is it a serous secreting, mucus secreting gland? Do you want to guess?
mucus secreting?
Mucus secreting, because the cytoplasm is pale for these cells. Okay, look at that. Okay, these are all columnar cells. Can you see it?
Mm-hmm.
Okay, so that is that. Okay, so this is a tonsil. So there is another good mucus secreting gland right here. Okay, so that is another kind of lymphatic tissue. Okay, there is a more diffuse kind of lymphatic tissue that you see in the intestine. Okay, so this is a cross section of the small intestine, and if you look at it, one side is thinner, the wall of the intestine is thinner, but on this side it's way thicker. It's almost 3 times the size of this side. Okay, so even if you look at this piece of intestine grossly, you will be able to see this thickening on one side. So when you go to gross anatomy and when you're studying the digestive system, look for this area. Okay, you'll be able to see a thick white structure that is extending throughout the length of that section of the intestine. So usually you see this in the ileum. Okay, ileum is one part of the small intestine, so you'll be able to see it in the ileum, and these are all
Gullet? Or briar patches? Briar patches?
what are these structures?
Are they Payer's patches?
They are the whole, yes, the whole thing is a Peyer's patch, but these structures are
Nodules?
nodules. Okay, so you see lymphatic nodules, so this whole thing is the Peyer's patch. Okay, Peyer's patches, and then all these are lymphatic nodules, and you will be able to see antigen presenting cells like right here in this region where you don't have villi on top. You know, these are all villi. These projections, we call them villi. They are specific to the small intestine, but where you don't see the villi, you're supposed to see some antigen presenting cells. Okay, so let's go down here. Okay, like right here where you don't see the villi on top. Okay, so because these are associated with the gut, we call this
Gullet?
GALT. Okay, gut associated lymphatic tissue. And if it's associated with the bronchi in the lung, you'll call it
Gullet?
BALT. Okay, and if it's associated with any mucosal surface, like here, you would call it
Malt?
MALT. Okay, that's as simple as that. Okay, so that is it for the lymphatic tissue. Do you have any questions?
I have a question. I have it written that microfold cells are in the epithelium where there aren't goblet cells. We can't see those, right?
Let me see. Where is it in your text? Is it in the immune system? GALT. Is it in the GALT?
Yes.
The lecture notes?
Yes. Or it was definitely in the lab notes.
In the lab notes, maybe. Give me 1 second. Let me go see. Can you tell me exactly where?
Um, it was under slide number 88, but I don't know exactly what page.
Okay, immune thymus. Let me go. Respiratory. Zero. No. Immune mod. Okay, GALT. B cell nodules. And anywhere in the lymphatic tissue, if you see nodules, you're going to see T or B cells in them?
B cells.
B cells will be in the nodules,
Mm-hmm.
and T cells will be in the diffuse lymphoid tissue. Okay.
Okay.
Where are we associated with Peyer's patch called intestinal villi, B cell nodules surrounded by diffuse lymphoid tissue. B cell nodule. The M cells, right?
Yes, those ones.
Specialized epithelial cells known as M cells. They endocytose antigen at these sites where there are no goblet cells. Okay, so for example, let's look at this here, Peyer's patch, right? So let's go take a closer look at this, at the villi. See, these are all the villi, right? You're seeing sections of the villi. These are the crypts, which are the invaginations of the mucosa, okay, going down. So all these white spaces are goblet cells. Okay, you'll see tons of goblet cells in the intestinal villi. But there are certain spaces like the one right here. Let's go. See, like right here, you see no goblet cells here, okay? And here is where you find the M cells. Okay, there'll be few villi at this location. All this is diffuse lymphatic tissue. These are all T cells, but here is the nodule at the bottom, which is made up of B cells, okay? And
So they deliver antigens, right?
so antigens would be delivered at this site, okay? And the M cells that are located here, okay, these are all epithelial cells. These are specialized cells. So they will endocytose the antigen and they will present them to the underlying B or T cells. Okay. And based on which cell is getting stimulated, there could be a T cell response where there are more helper T cells or regulatory T cells produced, or there could be antigen production by the B cells. Okay.
Okay, thank you.
So M cells can be seen only with specialized stains. You can't like call all of these M cells because they are there. You need to use specialized stains and then identify them. Okay?
Okay, thank you.
Okay. Any other question about the immune system? Okay, then we move on to the nervous system quickly. Okay, so here is the nervous system section of the, what is this?
The spinal cord?
This is a section of a brain. Okay, spinal cord is this one. Okay, so you identify the spinal cord by the presence of this butterfly shaped appearance of, what is this area called?
Gray matter?
Yes. So the gray matter looks like a butterfly. Okay. And all of this is white matter. So if you see this, then this is the spinal cord. Actually, this is upside down. So we have to actually turn this around this way. And I say this is upside down because I can see the ventral spinal artery here. Okay. So these are all spinal nerves. And let's see, yep, all those are spinal nerves. So here's the gray matter, all this is the white matter. Okay, so let's go to the brain. This is an earlier part of the brain, maybe in the midbrain region. Okay, So here is the cerebellar cortex. Okay. These are the fibers that connect the two cerebral hemispheres. Okay. This is called the corpus callosum. This is the hippocampus. Okay. This is the ventricular space, lateral ventricle. Okay. This is the third ventricle down here. Okay. And this is the ventral part of the brain. And what else do we have? You can see light and dark areas, right? So if you go to this region, let's zoom in a little bit. Okay, what is this outer zone called?
The whole thing? Or is it just the molecular layer?
The molecular layer, okay, is here. So there are different types of neurons here. Who are these guys?
They're neurons?
They are neurons. What type of neurons? They have a specific name, right?
Are those pyramidal?
Yes, these are all pyramidal neurons. Okay. And all that is because the neuronal cell bodies are present there, all this area would be gray. Okay. Gray matter. This is
White matter?
white matter. Okay. And then there could be alternating layers. So this could be, this is gray matter again. You can see neuronal cell bodies here. Okay. So let's go closer to look at these neurons. They're so pretty. So what is that?
Is that a granular neuron?
No. These are all hippocampal neurons, but like let's take this individual neuron. Okay, neuronal cell body. What is that? Dark structure.
Nucleus.
Okay, think again. This dark.
The axon?
Hmm?
Is it the axon?
No.
Nucleolus?
Yes. So this is the cell body. Okay. So there's the cytoplasm. Okay. All that is the cytoplasm. This is the nucleus. And can you describe the nucleus? Is it euchromatic, hyperchromatic?
Chromatic?
Euchromatic. So you're able to see a distinct nucleolus in there. Okay. The nucleolus is in here. Okay, so that is as far as the neuronal cell body goes. Okay, so all these are axons, okay, or white matter, all of this. So there are nuclei, like small cells associated with the white matter, right? See, look at these guys. Who are these guys? In the white matter, these nuclei belong to who?
Oligodendrocytes?
Yes, awesome. So these are glial cells, okay, that you see associated with the axons because they are going to be forming the myelin sheath. Okay, so all this is white matter right here, gray matter. So in the central nervous system, in the brain, gray matter is on the outside. Okay, white matter is more on the inside. Okay, so the white matter is made up of axons, right? This all that is bundles of axons running through the white matter, or yeah, running through the forming the white matter. So when we see bundles of axons running together in the central nervous system, we use a specific term for that. What is that?
Fascicle?
Fascicle is just a bundle. It could be a muscle fascicle, nerve fascicle, right?
Yeah.
But in the central nervous system, in the brain and the spinal cord, if you see a bundle of axons running together, what would you call it in the peripheral nervous system?
Ganglia.
Ganglia. So nuclei or tract.
Tract. Okay. So axons running together in the peripheral nervous system, in the rest of the body, you will see them as nerves, like your radial nerve, your ulnar nerve, right? Your median nerve. All those are bundles of axons that are running in the peripheral nervous system. So we call them peripheral nerves. But if there are axons running in the brain, we call them tracts. Okay? If we see aggregates of nerve cell bodies, let's go find one aggregate right here. Okay. Can you see these nerve cell bodies? Okay, see there is another distinct aggregation of nerve cell bodies. Can you see it?
Mm-hmm.
So you would call that what in the brain?
A nuclei?
Yes. See, this is called the nucleus. So there is, you have to make this distinction. There is a nucleus within a neuron, right? That we just call the nucleus. But this nucleus, an aggregation of neurons within the central nervous system, go by a specific name. You know, like for example, you have the paraventricular nucleus, okay, the arcuate nucleus. So it's nothing but an aggregation of neuronal cell bodies in the brain. Okay, so they have specific names associated with them. Okay, so you have to know the distinction. So let's go. There's one more structure that we need to see here. Okay, what is this structure here?
That is the choroid plexus.
Awesome. Okay, nothing but a bunch of capillaries that filters the blood. Okay, and you have something like plasma that is circulating inside and outside the brain, cushioning the brain also a little bit. Okay, so that is called the cerebrospinal fluid. The cerebrospinal fluid also runs all the way down the spinal cord. The central canal of the spinal cord, see like this guy here, carries cerebrospinal fluid. There is some cerebrospinal fluid that is around this space as well. Okay, just for cushioning, providing nutrition, removing waste, metabolic products, and stuff like that. So, okay, let's see. So we looked at the different parts. This is the front part of the brain, forebrain. Okay, now let's go to the hindbrain. Here you have the brain stem. Okay, is right here. And this is the
Cerebellum?
cerebellum. And what we call these?
Folia?
The folia. Okay, so arbor vitae is what we call it, the tree of life. So it looks like a tree, right, with branches and stuff. So here also, let's go closer to look at some of these neurons. Okay, here's the molecular layer again. Okay, neuronal layer. So this is gray matter right here. This is axon. So this is white matter in the middle. Okay, gray matter again. neuron, all these are neurons, smaller neurons, bigger neurons, molecular layer. Okay, so what are these neurons called?
Oh, the piriform.
Yes, piriform neurons or Purkinje cells, whatever you want to call them. And these neurons are called, these are the smallest neurons in the body, granule cell neurons. Okay, so these are also, these are present only in the cerebellum. Okay, and you see the axons running on the inside. Oh, there are some membranes that you have to know. Okay, covering the brain. Beautiful. Okay, look at these membranes. The one that is attached closest to the brain matter. This is called the,
Pia mater.
yes. Okay, let's go look at the spinal cord, then we can see the others. And this guy,
The arachnoid.
exactly, the arachnoid membrane. And then this would be the
Dura mater.
dura mater. So this is the pachymeninges. You have to know the pachymenix and the pia mater and the arachnoid membrane form the leptomeninges. Okay, you have to know that. What else do we have to know? Okay, so you know the gray matter and the white matter are kind of reversed in the spinal cord, right? So there's the ventral spinal artery, central canal. So let's zoom in to go take a look at this central canal. Okay, so this is lined by,
Simple columnar epithelial.
okay, what are these cells called? Ependymal cells. Okay, all these are ependymal cells that line the central canal. So let's go to this gray matter here. Okay, so that's the
Dorsal
what?
horn.
Dorsal horn. This would be the
Ventral horn.
ventral horn. Okay, so now we are going to look at structures of individual neurons first. Okay, before we go into describing. Look at that. This is a good neuron. Okay, so this is a neuron, right? This is a big neuron. There's another neuron, another neuron. There is also another neuron there. Okay, few more. So if you look closely at this neuron, let's go. So there is the
Nucleus.
nucleus inside. What is this blue stuff?
Oh, that is Nissel substance.
Yes, Nissl substance. What is it actually?
Ribosomes.
Ribosomes. Okay, so this is a very active cell. Okay, and it has these processes. What are these processes called?
Dendrites.
Dendrites. And then there is this process.
That
Okay,
should be an axon
I'm going to call that an axon. Why?
because it doesn't have any Nissl substance.
Yes, very good. So this place where the axon takes off is called the axon hillock. Okay, where it's taking off. So that's the axon hillock, no Nissl substance in this process. So we're going to safely call that an axon. Okay, some more neurons right here with a beautiful euchromatic nucleus and a nucleolus. All these are nuclei of
Oligodendrocytes.
glial cells to be safe. So you would call dendrocyte if the chromatin is kind of marginalized like these guys. Okay, and then this would be a microglia. Okay, because it has a teeny cigar shaped nucleus. Okay, but if you see a larger cell, okay, usually the oligodendrocytes kind of hang around neuronal cell bodies, okay, they tend to do that. But the astrocytes, you will find them around blood vessels, but those are the largest glial cells that you find, okay, among the 3 different glial cells. These are all microglia, these guys, okay, those are all microglia. Okay, so that is that. So what is this rest of this background called?
The neurophile.
Yes. Okay, so because we are in the ventral horn, right? What kind of neurons are these?
Motor.
Motor neurons. So if we were in the dorsal horn, then these neurons would be
Interneuron.
interneurons. Awesome. Okay, so that is that. That's all we have for the spinal cord. Then we have, what are these structures here? Give me 1 second. What is this?
Axons.
Yeah, these are all remnants of axons. Okay, and this space in life would be occupied by, why is it white now?
Probably something lipid related.
Yes, what is it?
Myelination.
Myelin. So in real life, all this would be myelin, okay, and that would be the axon. This is the remnant of the axon right here, but this white space would be filled with myelin, okay. So if there was a nucleus associated with this axon, it would belong to,
Myelinating cell.
which is a,
Schwann cell.
in the peripheral nervous system, you call it a Schwann cell. Okay, here it's still an oligodendrocyte because you're still in the central nervous system. Okay?
Okay.
Okay, so now let's go to. Where are we? Let me go to more nervous system. Okay. So you have to know the difference between myelinated and unmyelinated nerve fibers. Okay, so if you go to a section like this, a cross section, what would you call this?
Myelinated.
Myelinated. Okay, and the nuclei that are associated with this are nuclei of,
You said peripheral nervous system.
yeah, so peripheral nervous system. So these would be Schwann cells or neuronemocytes. Okay, so this pink stuff is called
Neurokeratin.
Yes, this is neurokeratin. Okay, so there is you have to know the difference between endoneurium, perineurium, epineurium. Okay, you should be able to distinguish this. This is a cross section. So in a longitudinal section, right here, this is how a myelinated axon would look. Okay, so this is with trichrome, right? So if it was, let's look at this, just H and D. See, this is how it would look in longitudinal section. So let's go closer to look at this. I'm trying to find the junction between two Schwann cells, like right there. There is one Schwann cell that is going, wrapping around the axon, another one starting off right there. What do you call that? This intersection.
The
There. The intersection of Schwann cells.
Node of Ranvier.
Yes, the node of Ranvier. Okay, see, nice looking nodes right here. Okay, that's the node of Ranvier. So this is a myelinated axon, right? So let's go here and take a look at this. So here is a cross section. So what would you call these?
Myelinated?
Yes, these are also myelinated, okay? Epinurium, perinurium, and endonurium, okay? You have to know all of those. And then let's go take another look at some neuronal structures right there. Maybe, oh, there they are. Okay, who are these guys? That one there, that one there, that one, that one.
Fascicles?
They are fascicles of myelinated or unmyelinated nerves.
Unmyelinated?
Unmyelinated because you don't see the neurokeratin. Okay, so what is this one right here? This is an aggregate of nerve cell bodies, but it's in the connective tissue of some gland. Okay, so it's obviously in the peripheral nervous system, the periphery somewhere. So what would you call this aggregation of nerve cell bodies?
Ganglia?
Awesome. So this is a peripheral ganglion. You can safely call this an autonomic ganglion. Okay. So this is in the periphery and it's associated with unmyelinated nerve fibers. So is nerve transmission, impulse transmission, faster in myelinated or unmyelinated axons?
Unmyelinated?
It's faster in myelinated axons. Because the nerve impulse can jump from one node of Ranvier to another. You see, in unmyelinated axons, the transmission is slow. Because when would you need like a lot of salivation to happen, right? Salivation is a slow process, right? You don't need, it's not like a fight or flight response that needs to save your life. So you will have unmyelinated fibers associated with it. Okay, so that is one thing. We need to look at one more structure. This is a peripheral ganglion, right? So this guy here, who is this? This is a special guy.
The dorsal root ganglia?
Dorsal root ganglion, awesome. What kind of ganglion is it? What kind of? Yes, these are all sensory neurons. Okay, are they unipolar, multipolar, bipolar?
Unipolar?
Yes, because their dendrites are out in the periphery, okay? And then the dendrites come here to the cell body and it goes towards the spinal cord as the axon, okay? The axon leaves this way. That is how it happens. These are all sensory ganglia, okay? So this is the dorsal root ganglion. So the nerve cell bodies are here, right? Let's go take a look. These are the nerve cell bodies, okay? And they have a nucleus, a nucleolus that is visible sometimes. Very nice nucleolus there, Okay, and these are all glial cells. We call them satellite cells. Okay, around these neuronal cell bodies. Okay, what else do we have? We did that, we did that. So that is it for the nervous system. Let's go to the digestive system. Yes. So we are going only up to the esophagus, okay? So let's look at the oral cavity here, okay? So this is a fetus, so this still has, what are these blue things?
Mesenchymal connective tissue.
This is mesenchymal connective tissue, okay? This blue is actually bone because this is trichrome, but this is developing bone, okay? We call that woven bone, okay? That is what this is. But this is a developing tooth, okay? But it all has all the cells associated with the tooth, nice and clear. So let's look at some of them. So this is the tooth, right? So that would be the red structure would be the,
Enamel.
yes, blue would be the,
Dentin.
yes. So the cells that produce the enamel that are present here are called
Amyoblast.
amyloblasts. Okay, so they have a E in them, amyloblasts, okay, just like the enamel. So the cells that produce the dentin are called
Odontoblast.
odontoblasts, okay, and they have a D in their name. So that is how you remember it, okay. So the enamel, what happens to the enamel when the teeth erupts?
The amyloblasts are gone.
Yes, the amyloblasts are gone, but the enamel is still there, okay. So the enamel provides an important covering for the dentin underneath. Okay, so this would later develop into the pulp cavity. Okay, so let's look at a full grown tooth right here. Okay, so this part is called the
Crown.
crown. This would be the
Root.
root of the tooth. Okay, this is the
Pulp ligament.
this is the gum. Okay, surface of the gum. This is the gum, right? So this is the pocket usually that we collect tartar in, you know, and all kinds of bacteria that collects in this pocket. And this structure is called the
Cementum.
mhm.
That's the pulp ligament then.
Okay, let me go closer. There. What does that look like to you?
Bone.
Bone.
That's
So
the
what bone is it? What bone?
Is it the jaw?
Yes, but it goes by a specific name. It's the jawbone, all right. So we call this the alveolar bone. Okay. So alveolar bone, that's the tooth itself. Okay. But we are down in the root of the tooth. Okay. So this outer surface or the root of the tooth is called the
cementum.
cementum. It looks very much like bone. Okay. Here you have osteocytes and lacunae. There you have cementocytes and lacunae. Okay. And it attaches, helps the tooth attach to the alveolar cavity by means of this
Periodontal ligament.
periodontal ligament. Okay. So here the cementum is getting wider and wider. See? Okay. And serves as a good attachment for the periodontal ligament and helps it attach to the alveolar bone. Okay. This is the
Pulp cavity.
Pulp cavity. And here you're able to see the
Predentin? Or the odontoblast?
odontoblasts. Awesome. Okay, there is the predentin. Here is the dentin tubules themselves. Okay, very nice, very nice. Okay, let's go. So that is it for the tooth. Okay, so this would be a typical brachydont tooth. Okay, this is a small tooth. You're able to fit it on a slide, but if it was a hypsodont, you won't be able to fit it on the slide at all. Okay, so here is a section of the tongue. Okay, so let's flip this around like that. So what kind of epithelium covers the tongue? What is this epithelium?
Stratified squamous non-keratinized.
Yes, but sometimes you can have keratinization, we'll see. Okay, what are these structures?
Papillae.
Papillae. Okay, and by the shape of these papillae, you can call them these are foliate papillae. Okay, because they look like leaves. If I have to show you a filiform papillae example, wait, let me see. Filiform papillae, where are you? Digestive system. Just for your, just for you to get an idea, okay? So this is how a filiform papilla looks like. Let's go. Oops. I don't know what happened. Got mad at me. Look at that. Still mad. See, look at those pointy things. That is how the filiform papilla looks like. Okay, lots of keratinization on top, but they are just pointed structures. They don't have any taste buds associated with them. Okay, so let's go back to our stuff for courses. Okay, where are we? Okay, let's go look at our tongue. See, in contrast to the filiform papilla, the foliate papilla have these structures. What are they?
Taste buds.
Taste buds, okay? So these papillae are capable of sensing taste, right? So we can call them gustatory papillae, okay? So let's look at the individual taste bud, okay? So it's made up of what?
You have chemoreceptor cells, the
Okay.
sustentacular cells, and the basal cells.
Yes. So there are basal cells that are basically stem cells that can differentiate into any one of these. And then you have neuronal cells, which are the taste cells, okay, chemosensory cells. And then you have the supporting cells that are the sustentacular cells. And the opening here is called the
Pore.
pore, okay? So that's the taste pore for you right there, okay? So these are all gustatory papillae. What is this structure here?
Salivary glands.
Yes, okay, so that's kind of emptying. So these are all ducts of the salivary gland, okay? More salivary gland here. This would be
skeletal muscle?
Think again. There is skeletal muscle in this picture. This is skeletal muscle.
Are those axons then?
Yes, of
The taste receptor cells?
taste receptor cells. I don't know. They could be, but in general, what would you call this bundle?
Bundle of axons outside of the central nervous system. Not a ganglia?
No. If it was a ganglia, you would see neuronal cell bodies with the nucleus, nucleolus, and all of that good stuff. This is just a bundle of nerves. I'm asking whether this is myelinated or unmyelinated.
Oh, unmyelinated.
Okay, unmyelinated axons. Unmyelinated nerve bundle is what it is. See, look at these. This is what?
Muscle, skeletal muscle.
Skeletal muscle, very good. Okay, this is longitudinal section, that's cross section, skeletal muscle. Okay, see more salivary glands, different types of salivary glands here. So same thing, if you go to this section of the tongue, here also it's going to be lined by stratified squamous epithelium. Okay, but you have a huge papilla here. Okay, this one has a moat around it, so it goes all the way around, and you can see it at the very back of the tongue. So when you're in gross anatomy, pull out your dog's tongue and look at the very back. You'll be able to see like 6 or 7 of these papillae. We call them circumvallate papillae, okay, because they are big and round. So are these gustatory or not?
Yes.
They are gustatory, okay, because they have these taste buds in them. Okay, same kind of cells. So all these are the neuronal, the axons coming from those sensory neurons. Okay, they are running down here, and they'll probably see they are like crisscrossing, probably connecting with an autonomic ganglion right here. Okay, can you see it? See more autonomic ganglion. Okay, another autonomic ganglion right there. This is a what is this structure?
Glands?
Yeah, this is a salivary gland also. Okay, salivary gland, skeletal muscle. So if we zoom out, let's zoom out. See, there is one type of salivary gland here, right? If it has all this red cytoplasm, then it's probably producing a watery proteinaceous secretion. Okay, but if it's paler like this one, cytoplasm is paler, it's producing a mucousy secretion. Okay, more salivary glands. You didn't think that this tongue would have that many salivary glands, right? Here, this one is more mixed. It has like mucus secreting and serous secreting salivary adenomias. What do you call them? Yep, secretory units. Okay, so more mucus secreting guys down here. See, things like that. But all this is skeletal muscle for you. Okay, so we are done with the tooth, the tongue. Then we'll go to the esophagus and we will wrap this up. Okay, so this is a tubular organ. So if it's going to be a tubular organ, it should have
Serosa?
serosa, adventitia, depending upon where it's running. But the structure, the wall structure should be divided into
The tunica.
tunics. Awesome. So tunica what?
Tunica lamina, tunica muscularis, tunica, or no, tunica mucosa,
Yes.
submucosa,
Yes.
muscularis, and then the adventitious / serosa.
Yes. Yes. Awesome. Awesome. So tunica mucosa is here, right? But this guy, because it's esophagus, it's going to be eligible for a lot of abrasive things going through the lumen. So you have what kind of epithelium?
Stratified squamous.
stratified squamous epithelium. So this is the lamina epithelialis. This would be the
Propria.
lamina propria, and this would be the
Lamina muscularis.
lamina muscularis mucosa. So the whole thing would be the tunica mucosa. Okay, this would be the tunica
Submucosa.
Yes, and this would be the tunica
Muscularis.
muscularis. Okay, so interestingly, some parts of this muscularis, let's look at it. Most of it is okay, like right here. Okay, see, all this is skeletal muscle. These bundles, but then these bundles, this is smooth muscle kind of mixed in with the skeletal muscle. It's weird. It happens only in the esophagus, I think. Okay. Particularly in this section, lamina muscularis mucosa will always be smooth muscle. Okay, it will never be skeletal muscle. But in the esophagus, there is some skeletal muscle in it because it can be voluntary, especially in bovines. Okay, when they want to regurgitate food, it has to be done voluntarily. So you'll have lots of skeletal muscle in the bovine esophagus. Okay, so depending on the outside, depending on whether there is a mesothelial covering on the surface or not, you can determine whether it is serosa. Like here, okay, there is mesothelium covering it, so this would be serosa. Okay, mesothelial covering, but here it's kind of torn. Can you see it? There is no clear covering. So this would be adventitia. Okay, what is this right here? Okay, come again.
There's going to be axons.
Of what kind of nerves? Myelinated, unmyelinated?
Unmyelinated.
Unmyelinated. So just call it an unmyelinated nerve bundle. Okay. So you have to keep, whenever you come across a structure like that, okay, just keep identifying it, just keep pointing out to it. You can see the, even from this zoom magnification, you can see there is a lamina muscularis mucosa here. Okay, so there is a distinction between the tunica mucosa and the tunica submucosa. But there is another part of this esophagus right here. Does this have a lamina muscularis mucosa?
No.
No. So what would you call this whole thing?
Propria submucosa.
Yes, propria submucosa, and all these are submucosal glands. Okay, again the epithelium is non-keratinized for the most part. Yes, it is non-keratinized. I thought it was keratinized somewhere a little bit, but maybe not. See, teeny bit of keratinization there, but it's not that big of a deal. Okay, so that brings us to the end of this session. Do you have any questions about the digestive tract or the nervous system? No? Okay, I'll let you go then. Okay, thank you.
Thank you
Thanks
so
so much,
much, Dr.
Dr.